Hi all, welcome back to my channel. I am Mahalakshmi JK. In the previous videos we have learnt about the introduction part, law of dominance and law of segregation. Now we'll proceed for the third law, that is law of independent assortment. This law of independent assortment is also known as dihybrid cross. It is called dihybrid cross because we are taking here two characters. One is the shape of the seed, that is round shape, and second one is color of the seed, that is yellow. And the second character, recessive character, we are taking as wrinkled seeds and green colored seeds. So this is our recessive character, and the dominant character is round and yellow seeds. Now for round and yellow we are resembling it as capital R, capital R, capital Y, capital Y, because these are dominant. For wrinkled green, which are recessive characters, we are resembling it as small r, small r, small y, small y. Now these are our parent pea plants. Now as we are doing the dihybrid cross, we should write them together. We are not supposed to separate it. Now the gametes produced are capital R, capital Y, small y. It is a dihybrid cross, so we are taking them together. Round and yellow RY, wrinkled and green small r, small y. Now these both have to be crossbred. Crossbred is nothing but a kind of cross pollination which you have studied in the sexual reproduction in flowering plants. So when they undergo for crossbreeding, they produce capital R, capital Y, small y. As R, two R's are there, we have to write them together. That is capital R, capital Y, small y. As R is dominant here, capital R, so it is round. And here capital Y we are getting, so it is yellow. So in the F1 progeny, we are getting the offspring as round and yellow. This is same as what we did for the law of dominance. Now once we obtain F1 progeny, we have to self-breed these. We have to self-breed the F1 progeny to get the F2 progeny. Now the gametes what we obtain are capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. These are the gametes of the first F1 progeny, and again we are considering F1 progeny, so the same gametes we are getting capital R, capital Y, capital R, small r, capital Y, and small r, small y. Now to obtain the F2 progeny, we go for the Punnett table method. Now in the Punnett table, we are writing gametes of one of the offspring in the row capital R, capital Y, capital R, small r, capital Y, small r, small y. And here in the vertical column also capital R, capital Y, capital R, small r, capital Y, small r, small y. Now when this is bred with this capital R, capital Y, capital R, capital Y, it will give us capital R, capital R, capital Y, capital Y. Now we have to write RR and YY together. We are not supposed to write RY, RY. Next is capital R, capital Y, capital R, small y. So it is RR, capital Y, small y. And the next is capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y, capital R, these two, small y, capital Y, capital R, small y, capital R, capital Y, small y, capital R, small y, small y. And in the last box again we are getting it as small r, small y, small y. Now we have to identify what gametes are these. Capital R, both are capital, so it resembles for the round. Capital Y, so it resembles for the yellow. Both are capital, again round. One of them is capital, one of them is small, but here the dominant character is capital Y, so we make it as yellow. Capital and small, but the dominant character is capital R, so it is round and yellow. Now after two more entries, here you can observe both are capital R, so we understand them as round. But here Y, both the Y's are small, so small Y, as both of them are small, they resemble for the green color, so I rate it as round and green. Now again, if we come here, one of them is capital, one of them is small, capital and small, so we directly go for the dominant character. If a single letter is also capital, then it resembles the dominant character. Again, if you can see here, both the Y's are small. When both Y's are small, it resembles for the green color. Now here, round and both are capital, so it is yellow. Now if you see here, till now we got everywhere one capital R at least, but in this case we are having both the R's are small, so small r resembles for the wrinkled character, so it is wrinkled. But these both are capital Y, so again yellow. Here both are small r, wrinkled. One of them is capital and another one is small, so it is yellow. Now I'll show you directly the last block. Both are small r, so it is wrinkled, and both are small y, so it is green, which is similar to the gamut that we have taken initially. Okay. So after filling the Punnett table, we obtain this kind of a result, which is our F2 progeny. Now we'll identify its phenotypic ratio, genotypic ratio, and their respective percentage. Now for the phenotypic ratio. So phenotypic means the appearance, how they are looking. So what different kind of phenotypes we are having here. First one is round yellow, round yellow, round yellow, round yellow. So everywhere we are having round yellow. Then next where it is changed, round green. Then round green is repeated. Then wrinkled yellow. And wrinkled green. Now just count here how many round yellows are there. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 round yellow. Then round green, 1, 2, 3. Wrinkled yellow, 1, 2, 3. And wrinkled green is 1. Therefore, the phenotypic ratio for F2 generation for a dihybrid cross is always 9:3:3:1. It is a fixed ratio for a dihybrid cross. Dihybrid cross is nothing but the third law, that is law of independent assortment. Now, the genotypic ratio, what all different types of genes are involved in this. So for that you need to observe carefully about the capital R's and capital Y's. Now the first one is capital R, capital R, capital Y, capital Y. First one is different from the second one because here it is small y. Capital R, capital R, capital Y, small y. Third one is again different because it is capital R, small r, capital Y, capital Y. Fourth is again different, capital R, small r, capital Y, small y. And the next one is, fifth one is similar to the second one, so I'm skipping it instead of writing. Next, sixth one is different. Capital R, small y, small y. The next capital R, capital Y, small y is similar here. And this is a new character. Capital R, small y, small y. The next one, it is repeated. Capital R, capital Y, small y is repeated. Then this is a new character. Small r, capital Y, capital Y. Then next, this is again repeated here. This is also repeated. This is new character. Small r, capital Y, small y. And the last one is totally different from all others. That is small r, small y, small y. As it is a ratio, just put the symbol of ratio everywhere. Now, how many capital R, capital Y are there? That is only one. The next one is capital R, capital Y, small y. On counting here, we are having for these two boxes. So the first box and the last box are unique. They are not equal to any other. Then these two are similar. Then the third box and the third box here are similar. And this complete one line is of the same gametes. So we come to the conclusion that capital R, capital Y, small y, this whole line is having that. So there are 4 number of this kind of a genes. And the next capital R, small y. Here we are having, so this is only one. Next is capital R, small y, which is here, which is similar to this box. So just remember this line. A box above this and a box above this, whichever I'm circling now, they are unique, and the corner two boxes are unique, and above circle and below circle are same kind. So it is 2. Then small r, capital Y, here it is, that is only 1. Then next is these two are similar, hence it is 2. And the last one is again 1. So the genotypic ratio is 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 is to 1. Now the percentage calculation for phenotype and the genotype. So in the previous video, I have told that the whole box is equal to 100%. And it is divided into how many boxes? 4 4s are 16. So it is divided by 16 boxes. Hence we divide it by 16. 100 divided by 16 will give 6.25. Therefore the value of each box here is 6.25. Now if I want to find for round yellow, as it is a phenotypic percentage, I am talking about directly the appearance. So how many round yellow we are having here? 9. So 9 into 6.25, 56.25%. Then if I want to find for round green, so how many round green are there here? 3. Hence 3 multiplied by 6.25 is 18.75%. Similarly, you can find for the wrinkled yellow and wrinkled green. This is how we find the phenotypic percentage. Similarly, you can find for the genotypic percentage also. Now, genotypic percentage, I'll show you just one. For capital R, capital Y, capital Y. Only one we are having here. So 1 into 6.25% is 6.25%. Similarly, you can find for the rest of the genotypes also. This is how we conduct the dihybrid cross. So just remember, law of dominance and law of segregation are but the monohybrid cross, and law of independent assortment is dihybrid cross because we are considering two different characters here. One is color of the seed and second one is shape of the seeds. So by this we conclude that in the law of independent assortment, along with the dominant and recessive character, there are some mixed kind of the characters as seen in the F2 generation. F1 generation is character, but F2 is having purely dominant, purely recessive, along with the some mixed characters like round green and wrinkled. So thank you for watching my channel. Please subscribe, share and like channel. Thank you.